Kia ora koutou katoa and welcome. Thank you for joining us for this session of the Thriving Cities webinar series. My name is Rachel Nepia and I'm the Program Manager for People, Cities and Nature, and I'll be your host for today. This series explores what it means for cities in Aotearoa, New Zealand to support truly thriving biodiversity and resilient urban ecosystems. Today's session is going to delve into the role of microbial communities as indicators and drivers of urban ecosystem recovery. Our speakers will share insights from their multi-year studies examining how microbial diversity, composition and activity shift through restoration trajectories in Aotearoa's urban centres. Our two speakers today are Dr. Kasey Kiesewetter and Dr. Craig Liddicoat. Kasey is a postdoctoral fellow at the University of Waikato. Her PhD research explored the restoration of soil microbiomes in Florida's Everglades, and now she's investigating microbial communities in New Zealand's urban environments. After Kasey, we'll hear from Dr. Craig Liddicoat. Craig is a postdoctoral researcher at Flinders University in Adelaide. His work uses DNA-based surveys to uncover the beneficial connections between microbes, soils, plants, ecosystems, and human health. After both presentations, we will have some time for questions, so please use the Q&A function at any point to send your questions through, and we'll get to as many as possible at the end. So first up is Dr. Kasey Kiesewetter.
Thanks, Rachel, for the intro and organizing the webinar series. It's great to share a bit about our work focusing on microbes in urban restoration. So I just wanted to start out by briefly talking about why we should care about microbes. So microbes are fundamental to all life on Earth. They're critical for ecosystem processes and services such as nitrogen cycling. They are necessary for plant and animal health and functioning, and they're also needed for human health. And today I'm going to focus more on the ecological side, and Craig will touch more on the influence of microbes and restoration on human health. So why consider microbes during restoration? We know microbial diversity and function are threatened by global change and human activities. And the interconnectedness of microbes with ecosystem dynamics makes microbial conservation essential for sustaining and restoring ecosystems. However, microbes have largely been overlooked in restoration efforts, especially in urban areas. So given this knowledge gap, an important first step to understanding microbial roles in restoration is to understand how restoration planting in urban areas impact soil microbiomes. And to do this, we surveyed 82 urban unrestored and restored sites across 9 New Zealand cities using a plot network created by Kerry Wallace in the first People, Cities and Nature program. Each restored site is embedded in a forest patch where the time since initial restoration planting ranged from about 6 to 70 years. And for each of these sites, we collected soil and then characterized soil properties such as pH, environmental data such as canopy openness, and climate variables like mean annual rainfall. We then extracted DNA from these soils to get microbial composition, and that tells us who is there, as well as microbial function, so what they're capable of doing. And I'm not going to go into details of these methods here, but I'm happy to discuss them or answer any questions related to the methods we used after the presentations if anyone's interested. So with this data, we first asked what drives community composition of soil microbiomes in urban forests. And before I get into the data, I just want to shout out Stella, who is a PhD who has been leading this work. So first we looked into restoration age because we know in other systems such as post-mining or post-logging sites, it can be an important factor for determining microbial composition. So to do this, we looked at how different the community composition of restored sites was compared to unrestored sites. So that's this dissimilarity here on the y-axis. And then we have time since restoration here on the x-axis, and we have a plot for each one of the cities. So if age influenced composition, we would expect to see community dissimilarity increasing with age. However, in our sites, restoration age has minimal, mostly non-significant impacts on differences in community composition. So next we asked, if restoration age doesn't explain differences, what does? So we found that local scale soil properties, specifically pH, soil moisture, and phosphorus, as well as climate variables, specifically rainfall, wind speed, and temperature, were all important factors for determining differences in microbial composition. So taken together, these results indicate two key findings within our system. The first is there's strong environmental context dependence on community composition. And second, local scale studies are likely not to accurately predict restoration outcomes across regions. And in addition to composition, we also wanted to understand what impacts microbial function in these restored sites. And today I'm going to just focus on one aspect of function, which is enzyme activity across 4 different functional groups. So we have carbon, nitrogen, and phosphorus metabolism, as well as microbial respiration. And on the y-axis, we have a set of predictors. And we tried for this study to focus on predictors that we might have some reasonable control over during restoration. So we mainly have vegetation and landscape factors. And we tested if these predictors influenced enzyme activity and whether they varied significantly from unrestored sites. So if these horizontal lines here don't cross this middle line here, it significantly impacted enzyme activity. And then our coefficient value on the x-axis tells us whether that enzyme activity was higher, which would be a positive value, or lower, which would be a negative value compared to the unrestored state. So if we look at microbial respiration, for example, we see that seedling richness, litter depth, and seedling number all significantly impacted microbial respiration, and that microbial respiration was higher compared to the unrestored sites. So from this, we can conclude that vegetation and landscape characteristics can impact microbial function. And this is critical because it means decisions that we're already making during restoration can matter for soil microbial function. But we still don't know what functional profile should be the target during restoration. However, this work is foundational to understanding what healthy or optimal soil microbiomes might look like during restoration. For instance, from this work, we can identify potential factors, processes, or trends that warrant further investigation that will help build restoration frameworks that support healthy functional microbiomes. And finally, we wanted to understand how forest succession impacts microbial networks. And microbial networks can be used to understand potential microbial interactions in communities. And to do this, we looked at centrality, which is a measure of a taxon's importance or influence in the network based on its connection or its position. And when we look at networks from unrestored and young sites, we found the highest centrality. And this indicates a greater interdependence among taxa and potentially less resilience to disturbance. As if we remove a key taxon, we could cascade that disturbance through the whole network. As time since restoration increases, we see a decrease in centrality, with these intermediately aged sites having the lowest centrality. So this indicates weaker or fewer potential interactions, as well as more transient taxa. It could also indicate potentially lower functionality, but it could also have higher resilience, as the elimination of a single taxon is unlikely to cascade to others. And finally, when we look at networks from the oldest restored sites, we see that centrality increases to an intermediate level, which could potentially indicate an increase in functionality compared to the intermediately aged sites, but more resilience to disturbance compared to the unrestored or young sites. And we were also able to compare the networks we created from our data to another study that collected this same type of data. And we found that the centrality in old growth forests had a centrality that was significantly higher than the oldest restored sites, but lower than the early restoration sites. So this suggests that even after 30 or more years post-restoration, networks from restored sites are still significantly different from old-growth forests. And I just wanted to end with a couple of key take-homes from our work so far. So first is that habitat and vegetation quality is critical for microbial diversity and functions. And given the heterogeneity across our sites, restoration time does not always result in high quality habitat. And this suggests that good restoration practices and decisions, as well as careful consideration of local and landscape contexts, especially those that are important for microbes, are likely to positively impact soil microbial biodiversity. And second is that time since restoration does impact potential microbial interactions, which suggests that time could be important for recovering certain interactions. However, it's important to note that restoration does not result in a return to pre-disturbance levels of centrality, which could indicate that in these older restored sites, there are either key connections that are still reestablishing or those communities are unable to recover them. And given that our oldest sites in this network range from about 31 to 70 years, it's possible that given more time, these communities could start to look more similar to old growth forests. But it's also important to consider the potential for more active microbial interventions that might allow us to aid microbial restoration. And this could be especially important for those intermediate aged forest sites, as this low centrality might give us an optimal stage to see the most impact for guided microbial interventions. And with that, I will hand it over to Greg.
Thanks, Kasey. Thanks, Rachel. Moving now to our study of the health promoting side of soil biodiversity in the context of ecosystem restoration. Microbiomes provide a tangible link between the health of ecosystems and the health of people. Microbes comprise a major part of us, and most of these ultimately come from the environment around us. Our microbiomes are critical for strengthening our immune system and shaping gut function and metabolism. We know that gut microbiomes impact our health because fecal microbiome transplantation, or FMT studies, show health protection and the transfer of disease. Human clinical trials using FMT are underway, aiming to treat a wide range of inflammatory, metabolic, and neurological diseases, bearing in mind that some level of evidence is required before moving to human trials. So microbiomes support human health in different ways, including boosting nutrition in our food, promoting gut health, and environmental exposures can also seed and shape our gut microbiome while supporting the wider field of One Health. That's the interconnected health of ecosystems, soils, plants, animals, and right through to humans. So what is the unit of interest for a healthy microbiome? It's not necessarily at the scale of an organism. There can be key beneficials or pathogens. There can be indicator groups associated with health or disease. Molecular patterns or bits of microbes' bodies can trigger immune responses for inflammation or triggering tolerance. Constituents of microbes' bodies can be taken up and used in beneficial processes. Also, microbial functions or beneficial metabolites can be produced through a string of activity or in extracellular space. So that's outside of the microbes' bodies using shared resources. And why focus on soil? Because it really is the richest of environmental microbial reservoirs. Estimates suggest that 60%, perhaps even up to 99.9% of species on Earth have a home or a sometimes home in soil. So that's really a lot of diversity. And some of those organisms will likely find their way into the human gut from time to time, many perhaps only transiently, some for a bit longer. In localized ecosystems, we see that restoration can shift microbial communities in seemingly predictable ways. All going well, you might see something like this, where bacterial communities with measures of community scale similarity to natural references on the y-axis show recovery over time in association with above ground plant communities, eventually reaching close to natural levels of variability equivalent to established forests. So as Kasey pointed out, the pecan data is spread across quite a large geographic range, whereas this type of neat outcome comes in quite a localized setting. So linking these ideas to human health, medical researchers and immunologists over the last 20 or 30 years or so have become increasingly confident that reduced exposures to natural microbial diversity through global changes such as urbanization and biodiversity loss have contributed to microbial deprivation and imbalances, driving poorer training of immune systems and increased allergies, autoimmune and chronic inflammatory diseases. Just for example, studies with daycare children have shown bringing natural soil and plant materials into daycare yards changes their skin and gut microbiomes and brings improved biomarker responses for more tolerant immune systems. Much of the work in the field has focused on immune-mediated effects, but there is a gap in examining potential overlaps and influence through metabolic effects. Globally, the areas of degraded land and soils are increasing. Similarly, the burden and cost of key metabolic disease, type 2 diabetes, is increasing at alarming rates. Type 2 diabetes is linked to energy imbalance, and there is an uncertainty over its origins. It has been linked to environmental influences that extend beyond just dietary factors. I've been exploring possible changes, possible overlaps in the functional profiles of degraded soil microbiomes and gut microbiomes in type 2 diabetes. This has largely used some model data sets, but I'll come back to the pecan soils data set shortly. I'm using a method that translates the functional potential of soil and gut microbiome samples to represent potential metabolism of individual compounds to indicate what the communities are geared up to process, more or less. So shifts could arise because of the food they are accustomed to eating and or the environments that the microbes have recently come from. In data sets from post-mining ecosystem restoration and type 2 diabetes from Sweden and China, I found degraded soils and type 2 diabetes gut shared increased potential metabolism for simple sugars, decreased potential metabolism for lignin or plant-based fiber, and decreased potential metabolism for monomethyl branch-chain fatty acids. Now, sugars and fiber probably make sense, but what are monomethyl branch-chain fatty acids? These are precious molecules known to nutritional science. They're key components of bacterial cell membranes. They're taken up and stored by fungi in the environment. And in humans, they're taken up to participate in host metabolism. So if this is the thing, what might be going on? Considering natural intact ecosystems on the left versus degraded ecosystem soils on the right, typically intact ecosystems have more plant biodiversity, fungal recycling networks, and pools of soil organic matter, which also release branch-chain amino acids. These ingredients may support microbes with higher levels of monomethyl branch-chain fatty acids in their bodies. Plant and animal-derived food from these high-quality ecosystems may also hold more monomethyl branch-chain fatty acids. With degradation, soil organic matter often breaks down to simple sugars, and reduced pools of organic matter and the branch-chain amino acids may mean microbes and food with less of those branch-chain fatty acids. If microbes from these environments are supplementing the human gut, then increased sugar metabolism may translate to increased energy harvesting. Reduced capacity to metabolize lignin may mean reduced production of short-chain fatty acids and problems with signaling gut hormones. If microbes and or food from degraded ecosystems are releasing less of the monomethyl branch-chain fatty acids into the gut, this could impact host metabolism through a number of ways. A key pathway may be that this type of molecule is needed to trigger a key mechanism of human host cell regulation. And when that's not working, it's been associated with diabetes, cancer, and neurodegenerative disorders. So did we find any of these signals in the pecan soils data sets? Using methods to account for variation due to the cities where we sampled, we found potential metabolism of the simple sugars generally increased in the younger, more degraded sites and decreased with age. Potential metabolism of lignin correlated with soil carbon and carbon to nitrogen ratios. And potential metabolism of the monomethyl branch-chain fatty acids decreased with higher levels of phosphorus and increased with high levels of moss ground cover. And I wonder if both of those signals with the phosphorus and the moss may be linked to fungal networks and recycling in the soils. Lastly, it's also increased with the fraction of nitrogen derived from organic sources. So there could be some crossover here with the pecan data. In summary, we know that ecosystem condition affects microbiomes and their capacity to process compounds, many of which are health linked. Soil and gut microbiomes and their functions have a degree of overlap, and that degree of overlap is yet to be fully defined. And a growing body of research tells us that exposure to naturally diverse soil plant microbiomes can provide a range of benefits, including potential to boost our immune system and gut microbiomes. So thank you. That's all I've got.
Thank you so much, Craig, and thank you, Kasey, also. That was incredibly fascinating. I really enjoyed listening to that. We are going to open the floor now to questions. So please submit your questions using the Q&A function, and I'll read them aloud for our speakers. I just wanted to get the questions started off with something for you, Craig, which is from the data you shared about the childcare centre and the exposure to microbes. Are we being too clean?
Yes, I think so I guess, from sort of hospital health scenarios, we're used to this very simple linear idea of a dose health response curve. It's a sort of a fancy term, but the idea that, exposure to too many microbes increases our risk of infection. Okay, so therefore we should really minimize that and just wipe away that risk and try and, have no exposure to microbes, if you like. And that makes sense in a hospital setting with immune compromised patients where you need to be clean. But out there in the real world, nature often gives us sort of non-linear relations, if you like. So like a U-shaped dose health response curve where, yes, if you're exposed to too much of any particular microbe, you're increasing your risk of infection, but not enough exposure where we're not getting the right training of the immune system. So being too clean can cause problems, whereas somewhere in the middle, we might get some, light sort of to moderate ambient exposure with all sorts of microbes, and the diversity helps control pathogenic behavior. But essentially, yes, I think we're out there in the regular world, we're too clean. A lot of the messaging, particularly through COVID, doesn't help with that type of narrative. But I think in essence, for the fitness of our immune systems, from an evolutionary point of view, we're used to working with a lot of different microbes.
So to get that kind of exposure that you need, is walking through the forest enough, or have you kind of roll around in the dirt?
Yeah. So like you're right. There's a whole range of a spectrum of different exposures, and even I wouldn't necessarily recommend going walking in the same forest all the time. So it's about diversity. So go walking through the forest, go walking through different types of green spaces, do a bit of gardening. Pets help. Even your diet, obviously, the diet has a major role because I guess some raw food will carry microbes on its own, but also your diet does help support or deplete your own gut microbiome just purely through the ingredients that are there and sort of promoting or fostering certain microbes to hang around. So diversity of exposures, a bit of soil contact, not too much, variety is the spice of life, so they say. So.
Cool. Thank you, Craig. Another question for you, Kasey. You mentioned that when you were discussing the differences in microbial communities between restored and unrestored sites, that we might need to look at interventions, particularly in the intermediate level of restoration. And I wondered if you could expand on that and explain what some of those interventions might look like. Don't forget to unmute, Kasey.
Thanks. So I think it is important to look at those interactions because composition doesn't necessarily mean interaction. But it's also important to note when we do networks, these are potential interactions. So they're actually not, we can't actually observe like microbe A interacting with microbe B. So they're not like true interactions like what we would see with like a pollinator network. They're kind of approximate or potential interactions. So we look at like how abundance varies, which is a bit different than when we look at composition, which is likely why we're seeing this trend. But both of them are kind of things we can use in looking at microbes to get an idea of like what the entire community is doing. So they're both good at kind of telling us a different piece of the picture. And then together we can kind of hopefully get more to a point where we have an idea of what our like target composition or what key species or potential interactions might be missing from these networks and communities in general.
So the kind of interventions that you talked about, would that be like soil transplants or
Yeah,
something
so
different?
it would very much depend on the habitat. I wouldn't recommend just like being like, I want to restore this and going and grabbing random soil and putting it in, because you could also transfer things you don't want. But there is a more evidence showing something called we call hub taxa in networks, which are these like key taxa or groups of taxa that have a lot of connections in the network. And they have been shown in the field for just the first time this year that they can help structure communities. So there's the potential for using these things like networks to say, hey, like if we want this community structure to resemble something else, we could potentially introduce some of those key taxa or groups of taxa into our networks. And it seems like in the intermediate ages, so this past this like just initial restoration, but before they start to mature into forest, it might be a really good time to do those interventions because those networks are really sparse, and they don't have a lot of connections, which means they could be a little bit more easy to manipulate at that point compared to like an older, more connected network.
Are you aware of anywhere in New Zealand where this has been tried?
No.
Interesting.
I don't. The study that showed that hub taxa are important for structuring microbiomes came out of Florida. So
Very interesting. We have a question here from Catherine about comparing restoration planting seedlings that are raised in commercial potting mix versus those that incorporate duff from under healthy trees of species such as a mother tree. Do you know of any studies around that, and does it make any significant difference?
So probably. I don't know any specific studies on the top of my head, but I know that anecdotally, there's a lot of evidence to suggest that microbes are really helpful with germination and like early plant development. Even just in my own work, my PhD, we found that we had a lot better survival and germination when we had plants with microbes compared to no microbes. We know that potting soil isn't sterile, so there is like a bit of a microbial community there. But when you have plants that have known interactions with key like fungi or bacteria, introducing them to these like juvenile plant stages could be really helpful in providing some sort of resilience, especially when they get moved out into the field and they have stress compared to a greenhouse where they're getting everything that they need.
Interesting. Was there anything you wanted to add to that, Craig?
There are different researchers trying different things to speed things along. I'm not fully across all of the different interventions that people are trying. I guess we were working with those previous sort of restoration efforts, so we haven't really studied that. But yeah, the other question in the Q&A was if I could just raise that for everyone, I suppose. What do I mean by exposure? Does the soil have to be ingested? And no, not necessarily, because there's mechanisms in the body where airborne dust and microbes, make their way into your airway, and there's mechanisms in the airway to sort of move that up and then to make their way into the gut. So even just dust in the air, soil on your hands, that is sort of interacting with your skin, your airway, and eventually some of it into your gut microbiome, just through natural mechanisms.
Awesome. Thank you, Craig. And thank you, Kasey. Also, your research, again, has been incredibly fascinating. So thank you for sharing with us. And thank you to everyone that joined us today. We hope this session has given you a bit of new appreciation for the role of microbes in urban restoration. Our next webinar is on the 25th of November and looks at urban forest soil biodiversity and restoration with Dr. Andrew Barnes and Dr. Kitty Wallace. We hope to see you there. Until next time, kia pai tō rā.
Thank you.